Hello, in this lecture, I will discuss phosphodiesterase enzyme and the drugs which inhibit it. This is an integrated lecture, therefore, there will be information from biochemistry, physiology also. Now, phosphodiesterase enzyme is a superfamily of over 100 different enzymes which are divisible into 11 classes. These classes are called as families. Each family has 1 to 4 subfamilies A, B, C, D. Now, these subfamilies are according to the gene coding. A number of variants are also found in each subfamily. Therefore, one typical example would be PDE1A3, and that will tell us this is a phosphodiesterase 1 with subfamily A and variant 3, and this is how it is expressed. Now there are various ways phosphodiesterases are classified according to their amino acid sequence, substrate specificity, and other characteristics. However, I will present here is substrate specificity and distribution of phosphodiesterase enzymes. Now, some of them are cyclic AMP specific. They will just inactivate cyclic AMP only, and these are the 4, 7, and 8 types, or family or classes. The second group is cyclic GMP specific, that is the class 5, 6, and 9. The third is dual specific, that is both inactivation of both nucleotides, and that includes 1, 2, 3, 10, and 11 class of the PDEs. Apart from this, one is cyclic GMP inhibited cyclic AMP hydrolyzing phosphodiesterase enzyme, and it is of class 3. So this is how it will be easier to understand a specific role of phosphodiesterase enzyme in these cells. Now, these enzymes are found almost all cells of the body, and here they regulate second messenger activities. And the enzyme is a hydrolytic enzyme, therefore it cleaves the covalent bond, that is a 3-5 phosphodiester bond between sugar and phosphate moiety of the nucleotides. These two nucleotides are cyclic AMP and cyclic GMP. They are converted into non-cyclic compounds 5-AMP and 5-GMP, and therefore they become physiologically inactive. Once their activity is terminated, the downstream events are regulated. For example, protein kinase A for cyclic AMP and protein kinase G for cyclic GMP. Activation does not occur further, so subcellular activities are not performed. Now, currently, inhibition of human phosphodiesterases is found to be a useful therapeutic strategy in selected diseases. And here later on we will know that some of the drugs are now actually in clinical use. Furthermore, research is being carried out to find out parasitic phosphodiesterases against which the drugs are developed to kill the parasites as anti-parasitic drugs, and the process is continuing. Now let us see the distribution, cellular tissue distribution of various phosphodiesterase subfamilies and variants. Now phosphodiesterase 1 has 3 subfamilies, and each subfamily has a number of variants. For example, A has 9, B has 2, and C has 5. Now phosphodiesterase 1 class is present in olfactory mucosa, brain, heart, kidney, vascular smooth muscles, lungs, and testes. That means tissue distribution is wide. Whereas phosphodiesterase 2 has only one subfamily with 4 variants. It is mainly found in brain, heart, adrenal cortex, platelets, lung, and liver. So the phosphodiesterase class 3 has two subfamilies, A and B. A has three and B has one variants or isoforms. It is again widely distributed in various body organs and tissues that include heart, kidney, blood vessels, hypothalamus, bronchi, fat cell, macrophages, T lymphocytes, oocytes, and pancreatic beta cells. At present, we have a number of drugs which are inhibitor of this class of the phosphodiesterase enzyme. Now, the class 4 has four subfamilies A, B, C, D, and each, for example, each has variants. A has 7, B has 4, C has 7, and D has 9. It's mainly found in the brain tissue, Sertoli cell, and lymphocytes. The type 5 has only one subfamily with four variants. It is located on the smooth muscles. Is an important thing. It's mainly located on the vascular smooth muscles, on the corpora cavernosa of the penis, on the bronchial smooth muscles, and also located in certain areas of brain and GIT. Type 6 has three subfamilies, and each subfamily has one variant. Now, this type is located mainly in retinal photoreceptors in the retina. Other sites are pineal gland and melanoma cells. Type 7 has two subtypes, subfamilies A and B. A has 3 and B has 4 variants. It is mainly localized in the immune cells. Immune cells also in muscles, heart, spleen, and brain. Type 8 has two subfamilies A and B. A has 5 and B has 6 variants. It is located on the cardiac cells, Leydig cells, adrenals, thyroid, and ovaries. So it's endocrine located and also on the cardiac cells. Type 9 has one subfamily and has 20 variants. It is located on GIT smooth muscles, lung, kidney, testes, and liver. Type 10 again has one variant which has one subtype with 6 variants, and it is located in the neurons and testes. And finally, type 11 phosphodiesterase enzyme has one subfamily with 4 variants. It's localized in prostate, testes, salivary glands, pituitary, and penile tissue. Now, genetic polymorphism is seen in the expression of phosphodiesterase enzyme. There are 21 genes that encode for various types and subtypes of family variants of the gene. For example, there are correlates which show that PDE4-D variant is highly expressed in schizophrenia. Similarly, PDE6A and 6B with amino acid changes due to mutation are seen in various types of retinitis pigmentosa. The variant PDE7B is found in cells of the chronic lymphocytic leukemia. PDE8B and PDE11A are found in Cushing syndrome and adrenal hyperplasia, and PDE4D is related with sensorineural hearing loss. So genetic polymorphism or the mutation of the phosphodiesterase enzyme may result in disease, or it correlates with the some of the existing diseases. Now let us come to the pharmacology of phosphodiesterase inhibitors. It must be understood that all phosphodiesterase inhibitors raise cyclic AMP and or cyclic GMP level, because some are cyclic AMP specific, some are cyclic GMP specific enzymes, so their inhibition will result in elevation of one or both of the nucleotides. Now there are two groups of the drugs currently available. One is non-selective inhibitor and the second is selective inhibitors. Among the non-selective inhibitors, methylxanthines are age-old medicines. caffeine, theophylline, aminophylline, diphylline, pentoxyphylline, doxophylline, and oxyphylline. Theophylline is a prototype, and this is a non-selective inhibitor having therapeutic utility as well as adverse effect profile because of elevation of cyclic AMP level. Now, selective inhibitors are tissue-specific uh, uh, drugs which inhibit tissue-specific enzymes. Therefore, they have selectivity of action and they will be more utilizable clinically in certain specified situations. This will be discussed later. Now, many selective inhibitors, although they are classed as selective inhibitors, but also inhibit the other subtype or subfamily of the phosphodiesterase inhibitors. For example, PDE5 also inhibits PDE6. However, it's more selective to PDE5 than PDE6. In a similar token, PDE1 inhibitors are also inhibitors of PDE6 enzyme. In fact, when we take an example of sildenafil, a number of the subtypes are inhibited apart from its specific action on the PDE5. Therefore, selectivity or inhibition, selective inhibition is relative. Now, let us see how phosphodiesterase inhibitors act. They inhibit the phosphodiesterase enzyme. For example, inhibition of phosphodiesterase 3 would result in no inactivation of cyclic AMP. It will increase the cyclic AMP activity, which in turn would increase phosphokinase A activation. Now, once it is activated, a number of things can be seen, effects can be seen. Myorelaxation, positive inotropic action, positive chronotropic action, cardiac stimulation, that is, decreased mediator release from the inflammatory cells, regulation of ion channels, hormonal secretions, and other secretions. This we will be discussing. In the same way, if there is inhibition of phosphodiesterase, for example, phosphodiesterase 5 enzyme that inactivates cyclic GMP, so there is no inactivation of cyclic GMP and there is increase uh, uh, level, and this increased level will increase the activity of protein kinase G, which is involved mainly in phosphorylation in smooth muscle. So the effect would be vasodilation, phototransduction in retinal cells, glycogenolysis in liver, smell perception in olfactory bulb, and apoptosis, that is cellular program death, which may occur in certain places. So this is how inhibition, whether selective or non-selective, will result in elevated levels of the cyclic nucleotide and activities. Now, taking example of non-selective inhibitor, theophylline, it inhibits phosphodiesterase enzyme without much selectivity. So the effect of the theophylline are multi-systemic effects which are due to cyclic AMP dependent and other actions. Now, among the cyclic AMP dependent actions are cardiac stimulation, vasodilation, bronchodilation, increased relief of anti-inflammatory mediator IL, interleukin 10, reduction in the diaphragmatic fatigue because it increases contractility. And some of the actually adverse drug reactions of theophylline, such as gastritis, headache, and induction of arrhythmias, are due to inhibition of the phosphodiesterase subtype 3 enzyme. So therefore, actions and adverse effects both can be explained by elevation of cyclic AMP. Additional mechanisms involved in theophylline action are anti-inflammatory actions, which is due to inhibition of the mediator release, and this is PDE4 inhibitory action. Some actions are not dependent on phosphodiesterase inhibition, and these are increased histone deacetylase 2 activity and the second is adenosine 1 antagonism that may explain the CNS stimulation and epileptogenic effect of the theophylline in high doses, and second is inhibition of the adenosine 2B subtype of receptors in the mast cells, and that reduces histamine release. I have not included details of theophylline, which can be the separate presentation. Now we will discuss selective phosphodiesterase 2 inhibitors. Now currently many drugs are being investigated. One is anagrelite. Now this agent is available for platelet reduction. This is called as cytoreduction, and currently it is approved for the treatment of essential thrombocytosis in a dose of 0.5 milligram twice daily. It not only inhibits phosphodiesterase 2 but also inhibits phosphodiesterase 3 and phospholipase A2, which is involved in generation of prostaglandins. Now the drug inhibits maturation of platelets from megakaryocytes, therefore number of platelets decreases. Now drug may cause adverse effects, tachycardia, dizziness, headache, and they are explained due to phosphodiesterase 3 inhibition. So there are those specific drugs to phosphodiesterase 2, but also inhibit other subtypes also. Now, drugs which inhibit phosphodiesterase 3 enzymes, the most important drugs are the inodilators, which are in current use for quite 2 decades at least. Amrinone, now called as Inamrinone, Milrinone, Enoximone. Now, the newer agent, which has multiple actions, is levosimendan. Now, this agent is calcium sensitizer, activator of ATP-sensitive potassium channels in the vascular supercells. Now, look at this. Whether amrinone or milrinone or enoximone or levosimendan, they all do two things. One is stimulation of heart, increase contractility, and increase in the heart rate. But the second is they cause vasodilatation. So this combination of vasodilatory action and ionotropic action is called as ionodilator action. Currently, they have been used in the treatment of acute and severe heart failure when other drugs are failing. They have limited utility because long-term mortality benefits are not approved. Now, other drug in this class, that is PGE3 inhibition, is silastazole, which has a predominant vascular action, not cardiac action. This drug is a potent phosphodiesterase inhibitor in the vascular smooth muscles. Therefore, it causes vasorelaxation. It has antithrombotic action because it inhibits platelet aggregation also. Currently, it is used in the treatment of peripheral arterial occlusive diseases, and it has also been tried in stroke. Now, if you take the typical example of inodilator inamrinone, by phosphodiesterase 3 inhibition in the blood vessels, it increases relaxation. The underlying mechanism of vasorelaxation is increase. It is very intriguing to know the same increase in cyclic AMP in blood vessels does opposite to what it does in heart. Therefore, it must be understood nicely. Increase in cyclic AMP reduces MLC kinase activity, myosin light chain kinase activity. There is reduction in the phosphorylation of myosin light chain, and so there is less activation and contraction, that is less actin myosin interaction, and thus myorelaxation occurs. So vasodilatory action is because there is less interaction and less phosphorylation. In contrast to this, when cyclic AMP levels are increased, there is increased phosphokinase A activity in myocytes, cardiac myocytes. There is phosphorylation of L type of calcium channels, which causes increased calcium entry. Other actions consequent upon increased cyclic MPR increase calcium release from sarcoplasmic reticulum with the availability of calcium. So calcium mediated calcium release. And the third is increased phosphokinase activity A will cause phosphorylation of the sites for sarcoplasmic reticula, which further enhances release of calcium through ryanodine receptors. So two ways there is release of calcium from sarcoplasmic reticulum, direct and through receptors. The same increase in phosphokinase A activity will increase MLC phosphorylation. In vascular blood cells, it is less phosphorylation or dephosphorylation. So this is how increased phosphorylation increases ionotropic action by binding of calcium with troponin C that allows actin to interact with myosin and causes contraction. So this is how at the intracellular level second messenger activity results in vasorelaxation and cardiac contraction. Now let us have a look at phosphodiesterase 4-Is inhibitors. It is considered to be the largest family with 4 subfamilies A, B, C, D. Each has a number of variants, so almost 27 variants are known for this uh, enzyme. Now, PDE4 is found in inflammatory cells and also in lungs, and therefore inhibition of PDE4 would have anti-inflammatory action. Selective phosphodiesterase 4 inhibitors. One drug is apremilast. Now this is a unique drug which inhibits almost all the variants and is currently approved for psoriatic arthritis and the dermal psoriasis, that is plaque psoriasis. Now basis of its use in psoriasis is that by inhibiting phosphodiesterase 4 enzyme, the drug decreases activity of the immune cells in this disorder because phosphodiesterase is found because phosphodiesterase 4 would cause greater concentration of cyclic AMP in the immune cells, and this will result in the reduced synthesis of pro-inflammatory mediators such as tumor necrosis factor alpha, interferon gamma, interleukin 17 and interleukin 23. It also stimulates the production of anti-inflammatory cytokines. One of them is IL-10, another is TGF, tumor transforming growth factor beta. So by these actions, there is strong anti-inflammatory action. Roflumilast is another PDE4 inhibitor which inhibits PDE4 in the lung tissue. It is the first approved drug for the treatment of prevention and treatment of exacerbations in COPD. As usually given, is a longer acting drug. It is given as 500 milligram once daily. Now that drug is found to reduce allergen-induced bronchoconstriction, and it also may induce weight loss as it also reduces appetite. The third drug is drotaverine, which is already established as antispasmodic drug. Its current mechanism of action is inhibition of phosphodiesterase 4 in the GIT smooth muscles, and that causes myorelaxation. It is perhaps the most commonly used drug for this condition, and it is commonly used alone or along with paracetamol and mefenamic acid in the treatment of colicky pains and spasmodic dysmenorrhea in a dose of 40 to 80 milligram 3 times daily. Now let us talk about phosphodiesterase 5 subtypes. Now PDE5 is cyclic GMP specific PDE. It has only one subfamily or subtype with 4 isoforms, and the gene for phosphodiesterase 5A is located on chromosome 4q27. Now distribution of the variants and subtypes are extensive in the body tissues and is located on heart, lung, liver, kidney, brain, urinary bladder, urethra, prostate, skeletal muscle, uterus, and penis, particularly corpora cavernosa of the penis. Now, PD5A3 is found in the vascular smooth muscles of the number of organs, including penis. So by inhibition of PDE5 variants 1, 2 and 3, there is vasodilatation, which we will discuss now. Now PDE5 expression is increased in neoplasm of breast, colon, bladder, prostate, lung and pancreas. The exact clinical significance of this finding remains to be known. Now what could be the potential therapeutics of phosphodiesterase 5 inhibition? Male erectile dysfunction is treated via inhibitors. Primary pulmonary hypertension of the severity moderate severity 2 and 3 is currently approved indication for that. Benign prostatic hypertrophy and male is an approved indication. It is being evaluated for premature ejaculation, female sexual arousal disorder, overactive bladder. Sometimes also thought that it will be useful as myorelaxation in renal colic, vasorelaxation in Raynaud's phenomena. It may prevent high altitude pulmonary edema and is currently investigated for protection of the organs, kidney and heart. So these are the potential uses of the phosphodiesterase 5 inhibitors. Taking the example sildenafil now. Sildenafil structurally resembles the purine ring of the cyclic GMP and therefore it competitively inhibits phosphodiesterase 5. Now, penile erection is initiated by non-adrenergic, non-cholinergic neural stimulation, which is much of the voluntary and controlled by higher centers. And once it comes in a thought, arousal, that releases nitric oxide from neurons and endothelial cells in the penile tissue, which in turn increases cyclic GMP. Now, cyclic GMP activates PKG pathway. So there is increased intracellular GMP which inhibits calcium entry into cell. Increased GMP increases PKG and activation of MLC phosphatase dephosphorylates MLC. Therefore, no contraction occurs. There is vasodilatation of corpora cavernosa which leads to erection intumescence. It is important to note that nitric oxide increases activity of soluble guanylyl cyclase, thus increases formation of GMP. So GMP formation is also dependent on nitric oxide in one way. Therefore, sildenafil will potentiate nitric oxide effect during sexual arousal, provided there is arousal. There is a desire for sex and therefore for sexual arousal is needed for his erectile activity. This is very important. Apart from sildenafil, at least 6 drugs are available in the same group for the treatment of erectile dysfunction and other indications. Sildenafil is a short acting, should be taken within 1 hour of the sexual act in a dose of 25 to 50 milligram. It is also indicated in pulmonary arterial hypertension. Tadalafil is somewhat longer acting and therefore should be taken once daily only within 1 to 2 hours of the sexual act, and it is also indicated in the treatment of pulmonary arterial hypertension and also in benign hypertrophy of prostate in lower doses. This drug may inhibit PDE11 also. Now, sildenafil effect is reduced with the food, but tadalafil effect is not reduced with the food. Vardenafil is another one, resembles sildenafil, and it is indicated for the erectile impotence only. Udinafil is also longer acting but less potent. However, it has a rapid onset of action. Avanafil is the latest addition, which is perhaps the quickest acting drug for the treatment of erectile dysfunction in causing penile erection, should be taken half hour before the act and it is as a longer effect. The food has a variable effect on it. Just to complete the list, a large number of other drugs are also promoted for the treatment of erectile dysfunction. Prostaglandin E1 analog, L-prostadil, papaverine, phentolamine in pipe therapy, aviptadil is a VIP analog, thymoxamine is intracavernosa alpha blocker, apomorphine works centrally, paroxetine and dapagliflozin are the SSRIs, selective serotonin reuptake inhibitors. They act centrally, and currently a new drug which acts on the serotonin receptors is a flibanserin. Now, flibanserin is called as female arousing drug, or in developed countries called as female Viagra. Sildenafil is available as Viagra, so it is a male Viagra, whereas flibanserin is called as female Viagra, and it is approved for the treatment of female hyposexual syndrome. Now, I have discussed type 2, type 3, type 4 and type 5, but there are other selective and non-selective inhibitors of other types which also have therapeutic utility or are being clinically evaluated. I will hurriedly complete up. For example, uh, type 6 is found, as I told you, in the photoreceptors in the retina, and zaprinast and sildenafil may also inhibit type 6. Sildenafil's adverse effects of poor discrimination of blue-green is because of inhibiting action on the type PDE6 enzyme. Now, type 8 is inhibited by a dipyridamole, which is actually antiplatelet drug with vasodilating action. Type 9 is found in the olfactory bulb, and currently only investigational drugs are available for the treatment of this, being evaluated in the treatment of sickle cell anemia. Type 10 is found in the penile tissue as well as in the corpus striatum, and papaverine and zaprinast are being evaluated as the drugs for erectile dysfunction. Type 11 is also found in many tissues including testes, prostate, pancreas and brain, and apart from tadalafil, investigational drugs are also being evaluated for the treatment of Alzheimer's disease and short and long-term memory. So this is how the development of selective phosphodiesterase inhibitors is being carried out for the therapeutic benefit in specific disorders affecting particular system. To summarize, the phosphodiesterase enzymes inactivate second messenger cyclic AMPA and GMPA and therefore regulate cellular functions. Selective phosphodiesterase inhibitors provide therapeutically useful drugs with distinct mechanisms of action and adverse effect profiles. Inhibitors of phosphodiesterase 3, phosphodiesterase 4, phosphodiesterase 5 have already found firm place in the therapeutics and are used clinically. More selective phosphodiesterase inhibitors of further isotypes are being developed for neuro and cardiovascular protection and for the treatment of genitourinary and inflammatory disorders of the respiratory system. So these are the references one can go through. Now I have almost completed the integrated lecture on the phosphodiesterase biochemistry, physiology, pharmacology and therapeutics, and thank you for joining this integrated lecture. The next integrated lecture will be on organophosphate poisoning. Good day.